Okay. So for those of you at home, I know it says part 3 of protein synthesis today, and we are not there yet. We are only to part 2 because, well, that's as far as we got. It's pretty complicated. I actually had a lab plan for tomorrow. I pushed that back to Wednesday. If you're at home, I would suggest that you look on Schoology and make sure you, I gave you kind of an alternate thing. So for everybody else that's watching this out here on YouTube land, thank you for your contributions. All right, part 2, amino acids are the building blocks of proteins. So if a protein is a train, the cars on the train are what, Matthew? If a protein is the train, what are the cars on the train?
The amino acids.
You're a genius, yes. We would call those monomers, the building blocks. What are the bonds that hold those cars together, Tay? They are covalent bonds. What type? Katie?
Covalent peptide.
Peptide bonds. Hence the name polypeptide. That's when you have a bunch of amino acids hooked together. It makes a many peptide bonds, which is basically the same thing as a protein. Some of those proteins are used to make up your body. You know, 50% of your dry mass is proteins. And then almost all of your enzymes in your body are made of those proteins. There are a few enzymes that are made from ribosomes, which we call ribozymes, but most all are going to be made from proteins. Part 3, proteins have to be folded in order to work. A good important thing to remember throughout this unit is structure equals function. And if it doesn't have the structure, it is not going to function properly. In the construction of the protein that we see at the ribosome, what protein structure happens there? You remember those different protein structures we talked about? Primary, secondary, tertiary, quaternary. What happens at the ribosome? Primary. And then you have the, remember what happens in the secondary structure? Making of 2 different things.
Isn't that like, it's just basically 2 polypeptides just kind of like.
Still one polypeptide.
And coiled, right?
Primary structure is when you add the amino acids together. Secondary structure is when you have the alpha helix or the beta pleated sheets. Either they're crinkly or they're twisty. That is done via what type of bonds? Hydrogen bonds. And then the tertiary structure is where it folds even more. There's quite a few things there, but one of the main ones we referenced were the disulfide bridges, which is another covalent bond. You have hydrogen forces at work, and there may be some ionic bonds as well. And then the tertiary structure, Walker, what happens there?
Tertiary structure is like multiple.
No. Tertiary is 3, sorry. Quaternary structure, my bad.
Quaternary structure, that's like multiple polypeptides.
Yeah, so you have multiple proteins that are hooked together, like in the case of hemoglobin in the red blood cells, where you have different hemoglobin subunits hooked together, collagen fibers under your skin where they're woven together. So definitely need to remember the 4 structures of protein. I think that's on page 80 or 81 in your book. All right, so we've already went through transcription in pretty good detail. Remember, transcription is what happens? Miranda? What happens in transcription? Mason?
DNA is turned to RNA or mRNA.
A segment of DNA is copied into mRNA. What do we call that segment of DNA? A gene. So a gene is copied into RNA. That segment, what is a gene, Miranda? Katie?
A part of the DNA that codes for something.
Codes for
Protein.
protein. So a gene is a segment of DNA that's going to code for a protein. Good. And that's copied into mRNA. And for those of you who didn't do what you're supposed to the last 2 days, that's on page 340 and 341. They go through the initiation, elongation, and termination using different enzymes and proteins. Primarily, the only one you need to be familiar with there is RNA polymerase 2. And they attach to the title box, which is important because you have to know where to attach and start copying. And then that segment of RNA goes to the ribosome. At the ribosome, translation occurs. So we've already talked a little bit about this, but we're going to get more in depth if you'll look on page 345 at the top right. Translation. This is where the protein is actually made. Part A, Walker.
In number 1.
Mm-hmm.
Okay. This process occurs at the ribosome, the translator.
Okay, so it's making it into something. Part B, Matthew.
The process turns the mRNA into a primary one, sequence of amino acids for making of the protein.
All right, when it says primary, it means that just the adding the amino acids together. So we just talked about the 4 structures of protein. This is the primary sequence. Remember, DNA and RNA is always read and made in which direction? 5 to 3, good. Remember that. Part C, Sarah.
This process needs the assistance of tRNA, transfer RNA, to transfer free amino acids from the cytoplasm to the construction site of the ribosome.
All right, where is tRNA made? Where is that something up here? Need some crackers. Where is the mRNA made? Okay, where's the tRNA made? Good. It's RNA, so where's it copied from? You're a genius, Mason. Genius, I tell you. Good. All right. So, in the responsibility of the transfer RNA, which look like little green inhalers in your book for reasons I don't know. I think the 9th grade book, they actually look little T-shapes, but whatever. So, their responsibility is to what?
Take the amino acids and bring them to the ribosome.
Yes, so it's getting amino acids from the cytoplasm, one of the 20 that it's specifically coded for, and it brings it to the ribosome to add on to that primary structure. Number 1 under translation, Tay?
Free amino acids are provided by the digestive system by catabolism, breakdown of proteins and food, and then delivered to the cell by blood vessels. Inside the cell, they are used for anabolism, building of proteins, or undergo deamination, removal of amine functional groups for ATP production and cellular respiration.
So this is a little bit of a review from the biochemistry earlier in the year. If you don't remember these words, then we need to have a talk. I'm a little worried about y'all's study habits, to be honest with you. But anyway, catabolism means breakdown of food. So you eat food, your body breaks it down. It breaks down the nucleic acids into... Nucleic acids get broken down into their monomer components called nucleotides. Proteins get broken down into
Amino acids.
carbohydrates get broken down into simple sugars. And then lipids, well, lipids can float around depending on what needs to be broken down or rearranged. So anabolism is that building back up. So we take, like, let's say we eat a protein. Our body's going to break it up into the 20 amino acids, and then our bodies are going to use those amino acids to construct the proteins that make us up or to make the enzymes that we use. Okay? Or, and the other option is, it can be used for energy in cell respiration. Remember how at the end of the cell respiration unit, we looked up how your body, primarily we looked at sugars, but your bodies can also use proteins or fats as a source of energy. It just basically has to cut off the amine group, that NH2, and then it turns it into ammonia, which you have to get rid of. We talked about getting rid of ammonia through urination and the importance of being able to get rid of those waste products. But anyway, that's the other option. If it's not being used to make proteins to build something or for enzymes, then it's being used for energy. Part D, 45 different tRNA molecules. We talked a little bit about this the other day. I know some of y'all were out. I don't know if you watched our explanations here on the tube. But if you turn to 339 real quick, I'll give you the simple version, which is the only one I can articulate. I'm not going to get a whole lot into this wobble effect or the Ionoscene because I don't think you're going to see a lot about this. But basically the idea here is, well, let's say look at serine in that second column from the left. How many different combinations of different codons code for serine?
4.
4. You can change you could have a mutation and have any different nitrogen base as the third nitrogen base, and what would happen?
So
You still get serine. So basically, this Ionoscene acts as a wild card, and I think it's kind of just stuck in on the end there. And so basically, it just makes it a quicker, faster process in terms of bringing amino acids to the ribosome. Now, really quick from the other day on that, let me go ahead and pass these out here. If you did not do this and turn it in, I'm going to be checking these pretty soon. So, did we finish this? Who was here yesterday or Friday? Did we finish filling that out or the things? The worksheet y'all had? Did we turn them in?
no.
We didn't turn them in.
All right, well, now I need them. Before the end of class. But anyway, let's see if I've got a few extras for you people who are on the lam.
I ended up finishing on this one.
Just for reference here. All right, and on the board here, I've kind of broken it up how I want you to do it. So you've got a segment of DNA that's running in the 3 to 5 direction. I want you to figure out what mRNA goes along with that. You got your thermo. And then use that to code for the protein that would go with it. Now, I've also added in tRNA right here. So, a couple things to take note of. You've got your DNA segment, whatever it is, okay? The first part of your protein is always going to be what?
AUG.
AUG. You've got to have the start protein. So that's not, I think, till maybe the third codon there. So you're going to have to look for that AUG. What's going to code for that AUG and mRNA? That is your start codon. It's still going to be an amino acid that you use, methionine. Methionine, yeah. Always tend to add in extra letters. It's still going to be that amino acid, but that's going to be where you start off. Now you can have methionine somewhere else in the strand, but it's got to be the start. Okay, so that's kind of like your capital letter at the beginning of the sentence. And then, okay, to make this clear, each 3 letters in the mRNA that codes for a amino acid, what do we call those? Codon. The 3 letters that match up with that, the complement on the tRNA to drop the amino acid off, we call the...
Wait, could you repeat that?
What are the 3 letters of tRNA that match up with the mRNA?
Anticodon.
Anticodon. So the anticodon and codon have to match up with each other in order to drop off the proper amino acid. Okay? Now, you will only use the mRNA codon to code for your proteins. Okay? The only reason I had you put the tRNA up here is so that you could see the complement. It has to be, the complement that has to be used with the mRNA to drop it off. But when you're using that chart on page 339, or if you have one on a test, which I'm sure you will, you need to make sure you're only using the mRNA codon to code for your proteins. Okay? Usually on tests, they have you do this with maybe 4, 5, 6 amino acids, and you have to code them out on the chart and then pick them out of a lineup or just leave them written out. So, it shouldn't be too hard. And then below that, you'll, like AUG, the amino acids, MET or methionine. Yeah.
So with that, so let's say we're doing we're starting with the start on top. All the stuff on top is what we're using to, I don't know.
Like for your template that you've been given, I would go ahead and code out all the codons, but the first one that's actually going to code for a protein is the one that's AUG.
Okay.
So you're going to have some before that aren't going to be, that's just going to be DNA prior to where that coding takes place.
Okay. And we're not going off this already given.
Yeah, you're using this as your template strand of DNA.
Oh, we are?
Yeah.
Okay, I did that wrong.
Well, how else would you do it? How else would you figure out your stuff?
Well, I mean, I used AUG as my, I can't really explain it. I guess I'll have to go over it.
I don't know. So it's in the 3 to 5 direction. So when you copy the mRNA template against it, the mRNA is going to be copied in which direction? 5 to 3. So, and then you should just be looking for that AUG as your first one. And then there are 3 stop codons. I don't have my sheet in front of me, but wherever the stop codon is, that's where you'll stop. Okay? Well, there's, I think, 3 of them. So any of those stop codons is going to be put a stop to your protein production. Does that make sense? These are the time for questions, people. Okay. So you need to make sure you make that protein, and then we'll go through and I'll check those. So that should be good practice. Hopefully by Wednesday, we've got our lab in for this unit, and basically what you're going to have is a bunch of cards around the room that have the codons and anticodons on there, and they have the whatever they're coding for, and you guys are going to have segments of DNA, so you'll code your segment of DNA, and then you'll go around and look for the proper amino acids that go with it. Except I think instead of actually making proteins, we're going to be using words. I don't know if y'all did this in 9th grade. Did y'all do that? Where you had the words on one side and you coded out the protein and you made a word? Yes? Okay, maybe Walker was the only one present that day. Who knows? All right. Let's see here. On D part, the anticodon matches the codon on the mRNA molecule, ensuring the correct amino acid is brought to the construction site. All right, so in 345, you can see that here. The codon that's in the middle is UUU, so the anticodon that would go pair up with that would be AAA, and it gets dropped off. And you're good to go. All right, let's see. For those of you, I think we discussed this with DNA, for those of y'all were out on Thursday and Friday, but if not, this whole 3' and 5', all that is where it attaches to the sugar, either the deoxyribose or the ribose. So when you see that 3 and 5, it's just talking about where that next molecule is attached. And it always has to go in that 5 to 3 direction, okay? Ribosomal structure. Who cares? 347, let's figure it out. All right. And just to note there on 346, they've got a 2-dimensional structure, like kind of like DNA when I just drew it out like a normal ladder, not twisted and all that jazz. Here's what that TNA structure looks like. It does kind of look like a T or a cross shape. And you can see it's just made up of a bunch of what? RNA nucleotides that's folded and bonded in a specific way. Does that make sense? Are y'all looking at the one at the top, the 2-dimensional one? You see how it says GCG, GAU, blah, 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 and the anticodon at the bottom? So all that is just a long mRNA sequence, okay? So, part 347 here. 2 parts of the ribosome. You can see it looks like 2 lumps of brown clay or 2 lumps of brown something. There's 2 different subunits here. I don't know that you need to know a whole lot about this. Subunit parts, they have to come together to make the factory. Here's what I will say. Ribosomes are made of 2 components. What are they?
Small subunit and large subunit.
Okay, well, what 2 macromolecules? What? All right, if you look at the top right there, they're made of proteins, which is the folded part, and they're also made of the rRNA. So this is your 3rd type of RNA. We talked about tRNA, we talked about mRNA, and then ribosomal RNA is the other component. So that's what makes up part of that ribosome. These are not membrane-bound organelles. So you know we talked about organelles at the beginning of the year, and we said, you know, when you're referencing an organelle in AP Bio, you're talking about membrane-bound structures that do stuff. Ribosomes are just little balls of protein and some rRNA, and they don't fit into that mold. Although they do have a very important function. Okay. All right. Let's see here. The different parts of the RNA. So for you, hold on, Katie and Miranda and Tay, y'all remember this? We talked about the EPA in environmental science. So this is the EPA of biology. You have, and actually it's really red in the other direction, so if you want to call it the 8, maybe that's better. On the ribosome, you have A is the what? What happens there?
Protein, or no,
tRNA gets added. So it's like the attachment site. Okay, so tRNA comes in, it's attached at the A site. The P site is where what's made.
protein.
The protein is made. Okay, the polypeptide is put together. And then on the E side, it's exiting. So it's pretty easy to remember. The attachment, protein making, exiting. And so that's where you can see the tRNA at the bottom there where it comes in, attaches, slides over, polypeptide's going to be added, and then it exits to go out and get some more amino acids. Okay? Any questions about the parts of it? They show you kind of an up close on 348 at the bottom there where the codon and anticodon have to match up. But I think we've got most of that. I laid in bed too early this morning. I've been better off to get up and come to work. All right. Parts of translation. Okay, hold on. On part E, read number 3 for us, Katie.
Number 3.
Mm-hmm.
The ribosome walks down the mRNA one codon at a time until it reaches the stop codon at the end of the mRNA molecule. Thus, having completed the message on how to make that particular protein. This walking is called translocation.
All right, as it moves down, the mRNA moves down the ribosome. Or excuse me, the ribosome walks down the mRNA. I mean, it kind of looks relative to one another there. Another interesting thing is you can actually put multiple ribosomes on the same mRNA. Why would you do that?
Make it go faster.
What go faster?
Translate.
Why would, okay, what would go faster though? If you put multiple ribosomes on the same mRNA,
Translation would go faster.
Protein synthesis.
you would make more proteins, right? That's what I'm getting at. So if you had multiple ribosomes going down through there, they're going to be making multiple proteins. So it's a way of making more. We'll look at what that actually looks like up close in a second. All right, at the bottom of 348, initiation. So these are the 3 phases. Basically the same as what we looked at in translation, where you have initiation, elongation, and termination. Initiation. All right, so the factory is built where everything can attach. And remember, methionine comes in, and that is your start sequence. And then don't forget about your APE sites where the tRNA attaches, protein is made, and then it exits. Elongation, page 350. During elongation, the actual making of the primary amino acid sequence. The ribosomes translates or walk down mRNA one codon at a time. Now, let me check the schedule here so I don't keep y'all too long. You might take a picture of it. Okay, you're supposed to know these things. All right,
I
let's see.
have one, sorry, I wasn't listening.
But you weren't.
We F lunch.
Yeah, so y'all don't get out here until 1:14. Okay.
Okay.
All right, so 1:14, somebody yell at me.
You get that? Yay.
You're gonna be hungry by then. Elongation. So you can see here how the ribosome is working its way down the mRNA, and how tRNA is coming in. We've already looked at this quite a bit. I'm not going to elaborate much more. What is it using for energy during this process of elongation as it walks down the mRNA? GTP. So that's our other energy molecule besides ATP that's used a lot. And then on number 3, termination, 351. The top here, termination takes place where a release factor enzyme comes in, starts hydrolysis. Basically, the ribosome breaks apart, the RNA comes off, and will then be probably recycled at that point, the RNA. Now, I got in an interesting conversation with someone yesterday about, I've got someone in my family who's a conspiracy theorist, I'll be honest with y'all. And they're convinced that the vaccine is, something's wrong with it. They're like, why does it have to be kept at 80 degrees below zero? Huh? What's the deal with that? And it's pretty simple, actually. Since both these vaccines are mRNA vaccines, I can't remember which one has to be kept at 80 below. I think the other one only has to be kept at like maybe 20 below. So I'm sure in their testing, they were looking at like what is the optimal temperature for this vaccine to work in the tests that they run. And so that poses a big problem because there's not many coolers that can keep vaccines 80 below anyway, especially with the slow rollout that Alabama currently has going on. I think we've only used like 1/5 of the vaccines we have in Alabama in the article I read the other day. But anyway, I said, why do you keep food in your refrigerator, Walker? Yeah, to keep it from breaking down. So mRNA, we've said all along, is very temporary, unstable compared to DNA. And so in a colder environment, enzymes and those particles are going to do what?
Slow down.
Move slower. They're going to break down much slower. And so basically it's just a way of maintaining that mRNA structure until you're able to inject it in someone. Okay? So don't be afraid. I'm sure there are a few side effects as with anything, but by and large, I think it's been pretty effective in the people that have gotten the vaccine. So, it's a free country, though. Do what you will. Who knows what they're doing in communist China? All right. Ribosome breaks apart. Number 4, the mRNA may be reused or it may be broken down and the nucleotides be recycled. Polyribosomes. Let's flip over to page 353. Now, on 352, they show you where this ribosome on the rough ER like secretes its polypeptide. We're going to come back to that. But on 353 at the top, poly means multiple ribosomes. So you have multiple ribosomes that attach to a strand of mRNA, and they move along that strand of mRNA. For what purpose?
Make the protein faster.
Yeah, you can make a bunch of proteins faster, right? If you've got 4 ribosomes that are copying or translating that mRNA code, then you can make a lot of proteins a lot faster if you put multiple proteins on there. And they show you some different electron micrographs of those polyribosomes to help in that process, okay? Post-translation modification. The protein is folding, must occur for protein to be functional. Remember, we've got primary structure, secondary, tertiary, so those involve some folding taking place. If the primary sequence enters a chaperonum, the protein will stay inside the cell. We briefly mentioned these earlier. It's basically a safe place inside the cell for a protein to fold. The entry is guarded by signal recognition particles, SRPs. If the primary sequence enters the rough ER, the protein will be exported out of the cell. Yeah, chaperone stays inside the cell, rough ER stays outside the cell. Let's look at the picture here on 352. So in this case, on the ER, that protein is, paying attention to that. That protein is made and it's secreted inside there in the ER. Where's it going to go from the ER? The Golgi apparatus, where it would be packaged and modified and packaged. A lot of times lipids are added, a lot of times sugars are added. That's where you wind up with glycoproteins or lipoproteins. And then it would be sent where? Out of the cell. Last thing, proteomics, excuse me. The study of genes and corresponding polypeptides made by those genes. So, if you want something fun to do in your time, there's you a side hobby. All right, 354 is kind of an overview of the whole process of transcription and translation. I would recommend that you look at that and read through it, and if you got that much, you know what's going on for the most part. Let's see. Beautiful. Part 3. Might catch up a little bit today. Remind me to stop at, what was it, 115?
114.
Whatever. Let's stop at about 110 and what time is it? 105 maybe, and we'll look at those data sets, and we'll look at the protein synthesis sheet y'all had from the other day that I think some of y'all may have had a little trouble with. Okay, part 3. Important concepts. Number 1, Tay.
A change in the nucleotide sequence is a mutation.
All right, a mutation is a change in DNA sequence. So the A, T, C's, and G's. Number 2, Katie.
Some mutations can cause cancer, abnormal growth, and organ.
Yeah, not all mutations cause cancer, but that's ones we hear about a lot because a lot of people get sick and die. Number 3, Miranda.
Prokaryotes would have, over millions of years, given rise to eukaryotes.
And we talked about the endosymbiote hypothesis, and I assume we'll refer back to that in a second. Types of RNA. So we already went through there. What are the 3 types?
mRNA, tRNA, and rRNA.
Good. And you really need to know like what each 3 of those is doing. So let's compare and contrast here. Prokaryotes and eukaryotes. And we've already discussed this briefly. Let's go back to page 337. All right, on 337, kind of our overview of this. Prokaryotes do not have what? Introns to be removed prior to translation. What do we say? As soon as you make that strand of mRNA, it can be sent to the ribosome for translation to take place. In eukaryotic cells, that is not the case. You have to modify that primary transcript and take out the,
Introns.
and do what with the exons?
Stitch them back together.
Stitch them back together. Part B, Matthew.
Genetic engineering, we can take 2 transcripts.
Okay. This is confusing the way of saying it. Let's just say secondary.
Secondary. We can take a secondary transcript out of a eukaryotic organism, use the enzyme reverse transcriptase to turn the mRNA molecule back into a DNA molecule. Insert the new DNA strand into bacteria. The bacteria will then be able to transcribe and translate off this new inserted DNA and thus make that protein. This has been done for numerous human medicines such as insulin or human growth hormone.
Okay, hold on a second. Here's a question you need to ask yourself. You might want to write this down in the notes. Why can we not use a strand of mRNA from a human and insert it directly into a bacterium for them to start making stuff? Why can't we take it out of a human cell, a strand of mRNA, and insert it in a bacterium directly and have them start making stuff?
mRNA is unstable.
Well, that's not necessarily the case because, I mean, everybody getting a vaccine is getting RNA injected into them. It is unstable, but it can still work. What's the bigger reason?
Well, DNA is the blueprint, and I mean, if you want to make more of those mRNA copies, then you would take DNA, and like it allows the bacteria to keep making proteins.
Okay, yes. So here we are, 412. I knew we'd get there. All right, well, let's stop on the way here at 367. 367 in your book. They go into a little bit more detail on the secondary transcript. Remember, you can't just take a piece of that initial mRNA because it would have introns and the bacteria would have no idea what to do with that crap, okay? It's like, what is this? It's like reading on a 3rd grade level, and then they give you War and Peace and like have at it, and you're like, my brain explodes. So, you can see the cap there, and then the poly A tail at the end, which we talked about, needed for protecting the mRNA until it gets to where it's going. Excuse me. All right, and then on 412 in your book, what I'm thinking. 412. Let's elaborate on this process. So let's say Katie is a diabetic, and she's not producing insulin, and her body is rejecting insulin that people are harvesting from pigs and cows and wherever they get most of their insulin from nowadays. And so we really need Katie to make some actual insulin that like Katie would have made if, her body didn't have autoimmune disease and destroy her pancreas. So we can take a gene for making insulin out of a human, whomever, should all be the same. And we can take that gene and get it into that mRNA form. Now, you could put that in a bacteria and it might make a little bit of insulin, but it's probably not going to make much before it gets broken down. So like Walker said, it's better if we take that gene and embed it into a bacterium. Why can we not cut a gene out of a eukaryote and embed it directly into a prokaryote? Introns. The bacteria would not know what to do with it. Now, I'll go ahead and say this. We have a pretty simplified way now of cutting and pasting genes out of stuff and putting it in other cells. So that's not the issue. It's what to do with the introns. So in order to get all the introns out, what do you have to do?
You have to let it go through normal transcript.
Okay, so you have to let it go through where you get to that secondary transcript. So you have a piece of mRNA that's going to code for a specific protein like insulin. And you take that mRNA, and then what do you have to do with it to get it back into DNA?
What is it? Reverse transcriptase.
Okay, so there's an enzyme, reverse transcriptase. There are quite a few viruses that use this enzyme because they are RNA viruses and they want to embed in your DNA. They use this same enzyme in this process to embed in your DNA. Like you think of like the herpes and a cold sore, like that virus is embedding its DNA in your DNA, and then when it gets agitated or cold or irritated or however you want to put it, then it starts replicating those viruses using that DNA that's already inside your cells. Okay? Basically the same thing here. So use that enzyme, it gets it back into a DNA molecule, and then that can be inserted into the bacterium. Okay? Now, hopefully in a couple weeks we'll have the transformation lab, and they already have the gene inserted for us because that's a little bit more than we have the capability of doing here in class. But we will take that gene in the plasmid where it's been inserted and try to get it into the bacterium and try to get them to act a little differently. All right, and then, talks about, Katie, quit rushing me. All right, part C talks about what we just talked about how you have to get rid of the introns. All right, so tomorrow, let's go and hit mutations and finish up part 3. Okay. So, what else say we needed to do?
We need to go over
The worksheet and what else?
data sets.
Oh, yeah. Yes, so the data sets I've told y'all were due, and so you should have those finished. Let's go over them so you have an idea of what's going on. I think I've got copies. Y'all look at them real quick after I pass them out. Remind yourself what they were about. One was listed on day 2, and one was listed on day 3. Alright, Mason, what happened the other night? Man.
That was sad.
What?
That was sad.
Do what?
That was sad.
I mean, like, I don't understand how the same people keep hitting the same threes over and over.
I mean, you shot 60% from 3.
Yeah, but once you see him hit like 14 threes from 25 feet, I mean, like, why not just get out on 25 feet and put a hand in their face? I feel like Coach probably said the same thing, right? We'll talk about it later off camera. All right. Data set number 1. Do y'all remember the picture in the Hershey Chase experiment?
Yeah.
Let me see.
Is that all FRQs and like data sets?
In here? No, there's a bunch of much more complicated and difficult stuff in here. We start getting into plasmid mapping and be like, it's okay, we're gonna figure it out, Katie. If anybody can do it, you can do it. I don't even know where this stuff came from. Sorry. I'm gonna tell y'all the wrong thing. Let me see.
I just remember as the picture with the blenders.
Yeah, I got it right. Okay, data set number 1. Not you. Alright, so this is the one on the Hershey Chase experiment, and the correct answer is, I think it should be C for you guys. Radioactive sulfur was used to track the amino acid cysteine. Remember, this is the one, the experiment where, they made radioactive, basically they made the proteins radioactive with one type of thing, the sulfur, and then they made the phosphorus radioactive for the DNA. And they were trying to see which one the virus was injecting into the bacteria so they could determine, is it DNA or is it proteins that make, is the genetic code for life? Because at this point in the mid-1900s, they didn't know. And so that's what they were doing. So radioactive sulfur is used to track the amino acid cysteine. Radioactive phosphorus was used to track the nucleic acids. And it showed that DNA is the ultimate source of inheritable information. It was thought by a lot of people at that time that it was actually proteins because they're so much bigger and so much more complex. So it was a bit of a surprise to find that out. And then data set 2, let me read this one out. Alright, the answer should be D. The transport protein would be made and inserted, but since the availability of cysteine is problematic, there may be fewer transport proteins. So there's not as much of it. You wouldn't make as much transport proteins. May lead to the cell not being able to control homeostasis sufficiently and causing the organism some ill effects. So there was a little amount of it, made little transport proteins, which may cause it difficulty in getting stuff in and out of the cell properly. Okay? And then, what was the other thing? The DNA thing, yeah. So, if y'all didn't get to this or figure this out, now's a good time to do it. If nothing else for practice, where you make your mRNA strand, what anticodon would go with it, and then what protein would code out. I forgot to tell y'all didn't really have to do all this coloring and pasting and stuff. I'm really just looking for the protein structure. So if you did that, though, congratulations, good job. All right, any questions?